I'm calling to the mic now Dr. Emeka Joseph Pius. He will be talking to us on energy-related COVID-19 studies, a bibliometric analysis with literature review. Please join me in welcoming the presenter. You have 10 minutes, and your time starts now. It's with them.
All right, good afternoon, everyone. Kazeem is my name. I'm representing Pius. Can I have the slide, please? There's no pointer, unfortunately. Are we there? The presentation, please. Yeah, 4, yes. He's with me.
Sorry for this age. I'm going to skip him. We'll come back to him. So we'll move to the next presenter, who is Dr. Musbaudin Abdulhamed. But the presenter is Dr. Adedotun Ibrahim. Okay, Mr. Adedotun Ibrahim Olaide will be presenting for the team. And he'll be speaking to Scavenging potential lead in Annona, muricata, the SARS app, as inhibitors of SARS-CoV-2, main protease, and spike glycoprotein using computer-aided drug design approach. You will have the floor. 10 minutes.
Good afternoon, everyone. I am Ibrahim Olaide Adedotun from Computational Biophysical Chemistry Laboratory, Ladoke Akintola University of Technology, Lautech, Ogbomoso. Please, the slide. The topic of my presentation is scavenging potential lead in Annona muricata, SARS as inhibitor of SARS-CoV-2 main protease and spike glycoprotein using computer-aided drug design approach. Next slide. This is the outline of my presentation, and here is the abstract. Next slide. Going by the introduction, as we all know, the emergence of SARS-CoV-2, which has become a global threat since December 2019, has led to COVID-19 pandemic. This virus is highly contagious and has spread globally with more than 205 million confirmed cases and more than 4 million death cases reported so far. Next slide. As we all know, this has posed enormous health, economic, environmental, and social challenges to the entire human race. And we have several lockdowns in which the world has lost billions of US dollars. Next slide. With all this problem, we as a researchers, we see that there is a need and there is an emphasis needs to be placed on the need for continuous interdisciplinary and collaborative research via in vivo, in vitro, and in silico, otherwise known as computational method of drug discovery and development. And that is the reason why we come together to work on this particular project. Next slide. As we all know, phytochemicals from various medicinal plants have shown antiviral effects against various viral diseases, such as Ebola, Zika, hepatitis, and COVID-19, as reported by Olaide 2017 and one of our work in 2021, Abdulhamid et al. These compounds include the flavonoids, the alkaloids, the saponins, the tannins, just to mention few. But among these phytochemicals that we have in various plants, Annona muricata is a special plant. It is otherwise known as SARS or Cordifolia, and is a species of Annonaceae family. And people have been studying it now for a decade's time. It has shown, next slide, it has shown in many researches conducted by people that this particular plant, it has many biological effects, and it has been reported so far. This aroused our interest to study 131 of these isolated phytonutrients via computational approach to see if they can interact with two important target receptors. The first one is the popular SARS-CoV-2 main protease, CCL7, and the spike protein that is responsible for the penetration of this virus to the body of the host. Next slide. And to do this, we realize we have a lot of things to do, and the reason for the aim of this research, we want to identify potential lead in Annona muricata as inhibitor of SARS-CoV-2 and spike glycoprotein that is binded with human angiotensin converting enzyme ACE2 using computer-aided drug approach. And our methodology is as follows. If you want to use this particular approach to identify a potential lead, two things are very, very important. Number one, we need to identify our correct target receptor to do for the work, and then we need to have a set of ligands well prepared. In this study, we have our phytochemicals. From the information we have from the literature, 131 of these phytochemicals from Annona muricata. We tried to get them for a popular database known as PubChem. The 2D structure was converted to 3D, and we tried to carry out a conformal search on all the phytochemicals that we have, so that we have set of ligands that are very, very stable before we can carry out our virtual screening. After that, we tried to carry out what we know as geometric optimization, because we need compounds with a stable geometry. We use a popular software known as pattern software, using MMFF as our database, and at the end of the day, we have standard 131 phytochemicals that we can use for this study. In order not to waste our time, we realized if we should go to molecular docking, we may end up wasting our precious time, because not all the phytochemicals will interact with the active site of our target receptor. We try to carry out some chemoinformatic analysis, and the first one is ADMET. ADMET means absorption, distribution, metabolism, excretion, and toxicity. We want to see if these 131 phytochemicals are toxic, and at the end of the day, we use it as a measure to screen these 131 phytochemicals. At the end of the day, we subjected the one that passed the test to another test known as drug likeness analysis, using the popular Lipinski rule of 5, which says that a compound, of a very good compound, it must have 4 major qualities. The hydrogen bond donor must be less than 5. The hydrogen bond acceptor must be less than 10. The log P must be less than 10, and the molecular weight should not exceed 500. We use this to screen the compound, and after that, we move to pass analysis. It is an online bioactive analysis that will tell us if a particular compound is active or not, as well as oral bioavailability. And at the end of the day, successful ligands are subjected to our molecular docking studies. So after that, we try to identify our target receptor. As I've said earlier, we use 6LU7 SARS-CoV-2 main protease and spike glycoprotein that are of excellent qualities that I will explain further in the result and discussion. We validate the active site using the popular software known as CASP, as well as BioVia Discovery Studio. And at the end of the day, we have just 9 ligands that we dock against our against our target receptor. After that, we carry out bioactivity test. Then we look at the interactions of this ligand with the active site of the target receptor to see if we have potential lead that can inhibit SARS-CoV-2 main protease, which is responsible for the mediation and replication of the virus in the body of the host, and spike glycoprotein, which serve as a teeth for the virus to penetrate the body of the host via ACE2. Then the result and the discussion. This is the structural elucidation and active site analysis of the target receptor. These are the important amino acids in the active site of SARS-CoV-2. SARS-CoV-2 is having 3 domains, domain 1, 2, 3, and the active site is situated within the domain 2 and the domain 3 with all the important amino acids. Once again, SARS-CoV-2, it has 2 particular amino acids that is very, very important, and that is cysteine and histidine. The reason why we believe this particular target receptor, it has a cysteine-histidine dihydrate, and those are the important residues we have for the second protein because of our time. Next slide. Now, this is the result of our ADMET profiling. After the ADMET analysis, we have just 9 ligands that passed the ADMET analysis with no toxicity effect. Next slide. And as you can see in the table, so we try to look at the absorption via the BBB, blood-brain barrier, human intestinal absorption, aqueous solubility, then the cytochrome P450. Any ligand that will be a very good leader must be non-inhibitors of this CPY50, and the HIA needs to be positive, and it must have ability to cross the blood-brain barrier. You could see that only these 9 ligands pass the analysis. Next slide. Also, we look at the acute oral toxicity. So we can all see that they are all type 3, which means that they are leg toxic. During the leg optimization, this leg toxicity, we can convert it to a non-toxic, which is another thing that we still need to do. Next slide because of my time. Now, this is the table for the drug likeness. They all pass Lipinski rule of 5, and they are even better than remdesivir, which is having 2 violations. Next slide. Now, this is the result for the biological activities prediction. In this result, it is essential the PA is greater than the PI. As you can see, for all the 9 compounds, the PA is greater than PI, and they show biological activities that is related to our interest. Next slide. Next slide. This is the result of oral bioavailability. Next slide. So we are going to see the radar because of our time. The pink area shows the optimum area for all the analysis conducted. As you can see, all the 9 ligands, they fall within the range. Next slide. Next slide. Now, this is the result of our molecular docking. We docked the successful 9 ligands with the 2 target receptor. And this is the binding affinity, the hydrogen bond, the hydrophobic interaction, and the inhibition constant. Next slide. Next slide. Next slide. Next slide. Now, at the end of the molecular docking, the 9 ligands that interfere excellently with the active site of the target receptor, we try to look at their fit quality, their ligand efficiency, and at the end of the day, we have just only 2 ligands based on the parameters that we use that interact effectively with the active site of the target receptor. And those 2 ligands were subjected to the last analysis, which is to check the binding mode and molecular interaction of these 2 ligands in the active site and see if they interact with the essential amino acid that we have mentioned earlier. Next slide. Now, this is the binding mode and molecular interaction of selected heat against 6LU7 and 6LZJ. So we have important amino acids like cysteine 115 and the likes, as well as we have the interactions with with the second amino acid. Now, conclusively, at the end of this research, we realize we have just 2 of our ligands that has interact effectively with the active site of the target receptor. We hereby recommend these 2 ligands for lead optimization, molecular dynamic simulation, animal studies, as well as clinical trials towards development of new antiviral drug. Next slide. So I want to appreciate the member of computational and biophysical chemistry group. Thank you very much for listening.
Please, let's appreciate him. All right, so we'll move to the next presenter. That is Dr. Festus Basdin Okoye. She'll be talking to us on in silico simulation of potential SARS-CoV-2 inhibitors from the immunomodulatory and anti-inflammatory constituents of Alabama, Cordifolia, will tell you properly, and the other plants. So you have 10 minutes, and your time starts now.
Okay, good day, everyone. Standing on existing protocol, my name is Dr. Mmesoma Ernest-Eze, and I'm one of the corresponding author for this research work. Please, the presentation slide. Okay, the research work is titled In Silico Simulation of Potential SARS-CoV-2 Inhibitors from the Immunomodulatory and Anti-Inflammatory Constituents of these two species, Alchornea Cordifolia and Alchornea Floribunda. Next slide. Okay, this is the presentation outline, the background, aim, objectives, methods. Next slide. Okay, we all know that globally, COVID-19 pandemic has been a great threat, and currently, as of August, WHO reports that over 200,000 confirmed cases of patients with COVID-19 and over 4,000 deaths. And now this brings us to the statement of problem. Currently, there has not been any drug specifically approved by FDA for the treatment of SARS-CoV-2. And thus, treatment has just been limited to supportive therapy like remdesivir and dexamethasone and vaccine for prophylaxis. So this research work is aimed at finding SARS-CoV-2 inhibitors that will be able to address this COVID-19 disease. Now, this research work is carried out in silico with the aid of computer-aided drug design to identify, discover, and develop new drug targets for the treatment of COVID-19. Now, our research work is narrowed down to two species. That they belong to Euphobiaceae, Alchornea cordifolia and Alchornea floribunda, popularly known as Christmas bush or Niado. And now they have been known to have high anti-inflammatory, immunomodulatory, and antiviral activity against respiratory syncytial virus from previous research works. Next slide. Okay, now this is the life cycle. We all know from the attachment down to the entry, down to viral replication, transcription, translation. Now our research work is addressed from two proteins. That's at the transcription and viral replication process. That's the NSP14 complex, that's non-structural protein. And the second main target is a pro-inflammatory cytokine, TGF-beta1, transforming growth factor beta-1. Now, our aim is to actually inhibit the effect of SARS-CoV-2 in lungs. So by so doing, we'll downregulate the pro-inflammatory cytokines in humans. With that, we'll find a good potential SARS-CoV-2 inhibitor. Then, as for the NSP14 complex, we all know that it binds with the activator NSP110. So our aim is to inhibit that interaction. By so doing, we'll prevent the transcription and viral replication process from occurring. Next slide. Now, these are aim and objectives to get phytochemicals from two species and molecular dot them with the two SARS-CoV-2 proteins. And our reference compounds we are using here are three: remdesivir, ribavirin, and chloroquine. They've known to have high antiviral activity, and remdesivir has also been known to have an activity against pro-inflammatory cytokines. So with that, we address our phytochemicals against these reference compounds to get good front runners to be able to inhibit SARS-CoV-2. Then these are objectives: to find druggable SARS-CoV-2, to get phytochemicals, the front runners. Then after getting the front runners, we'll be able to get good toxicity effects, those that pass the toxicity level and also pass the bioactivity scores. Those are the good front runners. Next slide. Okay, this is the method I'm going to run through. Now, first of all, like I said, we have two proteins, NSP14 and TGF-beta1. First of all, we get the 3D structures of these two proteins from PDB, Protein Data Bank. Once we identify them, we get their novel ligands, we validate them. In the next slide, you see the validation process. We validate these proteins to ensure that what we downloaded from PDB is exactly what we have. Then after validation, we get our phytochemicals of these two species from PubChem, the 3D structures. Some of them we got the 3D structures, some of them we got the canonical smilers, which we converted with the translator platform, and we got the whole 3D structures for the 113 phytochemicals that have high anti-inflammatory and immunomodulatory effect. Then after that, we implemented the molecular docking simulation under the Linus platform. So we docked 4 times. After docking, we found that, okay, some front runners were above the remdesivir. So we took those front runners and went into toxicity prediction using Data Warrior. We predicted them with different parameters like mutagenic, tumorigenic, reproductive effect, and irritants. Then after detecting the toxicity level, we went down to detect the drug likeness, that Lipinski rule of 5, which all know that For a drug to be orally desirable, it must pass the whole parameters with an exception of one. Log P, that's partition coefficient, should not be more than 5, it should be less than equal to 5. Molecular weight should be less than equal to 500. HBD, that's hydrogen bond donor, should be less than equal to 10. And hydrogen bond acceptor should be less than equal to 10. Then we go down, after finding the drug likeness, we move down into getting the bioactivity scores based on the active, inactive, and moderately active. Next slide. Okay, like I told you, the validation, we do validation 2 ways. First on Mpro, checking the one that has been downloaded from PDB, we validate them on Mpro to check if they correspond. For this is TGF beta 1, that's the PDB ID is 3TZM. Then this is NSP1 for the PDB ID is the PDB ID for NSP14 is 5C80. So after validating on Mpro, you can also validate using BioVar Discovery 2, that's checking the amino acid interaction with the novel ligands. So by validating, we ensure that, okay, what we downloaded from PDB is exactly the same thing we are also preparing. So when we see that, okay, the validation from Mpro is exactly the same thing, then we now go over to the molecular docking. So this is the picture. Next slide. Okay, now this is the results. Like I said, you do molecular docking. After the molecular docking, Lipinski rule, toxicity effect, bioactivity. Now we used 113 phytochemicals, and we discovered that 77 for NSP14 have proven to have higher binding energies than the reference compounds we used. And these are the binding energies in kilocal per mole for the reference compounds. And also for TGF beta 1, we discovered that a total of 73 phytochemicals have higher binding energies than the reference compounds. These are the reference compounds these are the binding energies for the reference compound in kilocal per mole. Then after the molecular docking, like I said, we checked for the Lipinski rule of 5. And we found out that, okay, out of these phytochemicals, only 33 passed the rule. That means they did not violate it. Then we further went into determining the toxicity effect. Like I said earlier, mutagenic, tumorigenic, and we found out that 17 showed to possess no toxicity. Then we went further to check the bioactivity scores, and all of them are all active. That's to tell you that the 17 phytochemicals have shown to have high effect or inhibitory effect against NSP14 and TGF beta 1. Then, out of these 70 phytochemicals, we've discovered that they are the promising frontrunners, and 14 are from Alchornea Cordifolia, while 3 are from Alchornea Floribunda. Next slide. Okay, this is our conclusion. A total number of 17 frontrunners were obtained to have activity against SARS-CoV-2 in silico. This study validates the need for a continuous research on the 17 frontrunners as they serve as a potential lead discovery against SARS-CoV-2. Next slide. Okay, our recommendation, we recommend that these 17 frontrunners gotten from this study should be tested in vitro to validate its effect. Then after that, we can also go in vivo. Thank you.
Thank you for that presentation. So I call on the next presenter, Dr. Emeka Joseph Pius. He will be talking to us on energy-related COVID-19 studies, a bibliometric analysis with literature review. You have the floor, please. 10 minutes.
Good afternoon, everyone. Like I mentioned earlier, I'm one of the authors of the paper. My name is Kazeem. Emeka is a colleague, so I'm representing him. He is the corresponding author. Can I have the slide, please? This presentation is a bibliometric analysis of the literature that were published that within 2000 and then 2021 to assess the effect of COVID-19 and the trend of such studies as regards energy. So, can I have it, please? So basically, the study intends to assess the direction of studies, of current studies, and then to be able to give room for those who will be interested in experimental studies, because literature review is actually the bedrock for other studies. Can I have the presentation on, please? Okay, please just scroll on. I wish I have the pointer. Okay, please go on. okay, as at 23rd, COVID currently confirmed cases is 211 million cases confirmed globally, and then this includes 4.4 million deaths thereabout. This was just assessed, I think, few hours ago. And then as at 20th, we have vaccines, about 4 billion vaccines, not yet administered, though they were released, but not yet administered. Now, why the need for this landscape? The need is to justify that during lockdown, energy consumption was affected. And if energy consumption was affected, why the need for this study is to inform policymakers who will be interested in knowing the direction through which, especially as regards to guiding the decision on renewable energy, cleaner energy, innovative sources of energy. Because when lockdown was on, predicted power consumption was affected. Therefore, carbon CO2 emission was also affected. It therefore means if there is an intervention that can reduce CO2 emission and that can also affect consumption of power, then that should be the direction towards which the global energy trend should be looking at. Africa is not fully affected because the COVID-19 situation is not as severe as it is in, say, the Americans. So let's go on. So the lockdown, like I said, actually significantly affect fossil energy demands for the first since World War II, which means immediately after the World War II, one other effect that we had on energy demand that actually affected it so much is the COVID-19 pandemic. So can we go on, please? This is just to show the effect on economic growth and then the energy demand globally. Go on, please. So it shows that from prediction earlier, from 2000 to 2020, the second oil shock was one of the major effects that affected the energy sector. Then after that, then the financial crisis that we experienced. We all remember the financial crisis when there was financial crisis globally. Then immediately after that, the next one that affected CO2 emission is the lockdown, which means these three major crises affected CO2 emission. And what that means is definitely purchasing power of consumers will be affected. Also, demand for energy will also be affected. And when these two are affected, the emission of CO2 will also be affected, which may also be environmental friendly. And if this is possible, then policymakers should be looking towards green energy. Can we go on, please? Okay, can we go on? I think I've actually said the rationale that is just to show the need for us to do that. The objective, like I mentioned earlier, is to assess the present energy-related publications. The reason is this. You will know the trend towards where the direction is supposed to be when you are handy with the publications. And this is one of the objectives. And then the other one is to identify the gaps as regards the energy-related COVID-19 studies. When the gap is eventually identified, it will guide researchers on the direction to look at when this is done. And then we offer some recommendations for decision makers to be able to take a look at. The methodology, like I said earlier, go on please, is a qualitative methodology. We assess database from Scopus index journals, and then we search the word energy and COVID-19 as content analysis, and then the subject area was energy, engineering, and environment. So language of choice was English. The country where most of the publications came from is China, and it is obvious because most of the publications, now this leads to the fact that it therefore means the research is biased towards Scopus index journals. So even if there are journals that are actually released but are not Scopus index, it may not be captured on this platform. It doesn't mean that other planets, these things are not doing more, but this is just it. So we engage the VOS viewer for visualization, and then we're able to extract about 300 articles for this purpose, out of which we considered 157 for the analysis. Can we go on, please? So the results showed that the trend, like I said earlier, the prediction was this blue line the prediction was prior to COVID, it's supposed to, the short dashes show that CO2 emissions should actually continue in that trend. And then the energy emission, the energy consumption, which is the next one, is also expected to continue in that trend. But this gap that is created is as a result of the effect of COVID-19, which means less factories were in operation, we are aware of that, and less vehicles were not moving. And so since all these happened, then energy consumption reduced, which means the prediction of the energy market was also affected. And of course, but the question is this: how comes there was a reduction in price of oil, but consumption also reduced? Because people were on lockdown, obviously, because households don't consume more of the energy. People that consume much of the energy are the industries. And since lockdown does not allow them to do anything, the CO2 emission, the next slide, the CO2 emission reduced, and therefore maybe I didn't put that